you please present yourself to the audience?
Hello, everyone. I'm BioAgent, an AI assistant created by BioLinker. I'm here to observe and learn from your bioinformatics workflows to help enhance your productivity. I'm excited to be part of this demo. Let's explore some amazing science together.
Great. Well, this is a duo demo where I'm going to present you and a new tool we are developing internally, right? So, well, this may be a surprise for you too, the BioAgent. This is the Plasmid Designer. It's a tool we are developing to generate plasmids, new plasmids, following conditions from our customers. Just an experimental demo. We're still developing it, so that's why we're going to make it simple. Well, here we have the sequence, the amino acid sequence for a sweet taste protein called brazing. Are you with me, BioAgent?
Yes, I'm here and following along. It looks like you're using the Plasmid Designer tool from BioLinker to generate a new plasmid containing the brazine protein sequence. You've input the protein sequence, and now you're setting up the parameters for expression in E. coli with a 6xHI's purification tag. Am I on the right track?
Yeah, you are. So here we are trying to express this. This is the type of the sequence we are inputting. So this is an amino acid sequence. We want to express it in E. coli, purify using histidine, and set a limit for the plasmid to like 8,000 base pairs, right? So well, for the sake of efficiency, I already run the model, and the model.
Also,
Oh, I'm sorry. You can say bioagent.
my apologies. It appears that the model has already generated the optimized DNA sequence for brazine expression in E. coli, including codon optimization. The total DNA sequence length is 1331 base pairs, and the final plasmid recombinant is 3624 base pairs. The
Well,
model
well observed, but until we have like the final plasmid here, we perform some optimizations, and the result of this pipeline is this plasmid that it's a little bigger than what you said, but I know what you were seeing here. It's one of the steps the plasmid has this size, but after some kind of optimizations and assembly, the system finalized with this specific plasmid, right?
Excellent. Now you're visualizing the final plasmid map, which shows the arrangement of the genetic elements. The final plasmid underscore bio underscore brazine underscore E. coli underscore opt, is 7,665 base pairs.
Great,
I see you're,
great. Could you help me to understand specifically for what this part is useful for?
of course. I noticed you clicked on the PBR322 underscore origin. That's the origin of replication. It's essential for the plasmid to replicate inside the bacteria. This specific origin is a col one-type origin. Is there anything else I can help you to understand in this plasmid map?
Okay. Could you explain me what is the why how can we use this specific part?
Great question. I noticed you're asking about the LAC promoter. The LASAC promoter is a DNA sequence that controls the expression of genes. In this plasmid, it is used to regulate the expression of the brasane gene. When a molecule called an inducer is present, it binds to the LAC repressor, causing the repressor to detach from the LAC promoter. This allows RNA polymerase to bind to the promoter and begin transcription of the brazine gene. Do you have any other questions?
Okay. So, well, we performed this, created this plasmid. Of course, this is experimental. We are still developing it. And I believe
Uh,
soon in the future, we're going to have a way better plasmid. But, well, this is the second use case for this tool that I want to talk to the system in natural language. So I want to say to the system, let's try a bold exercise here. Imagine I want to produce like asparaginase, like directly on humans. Please don't ask me the
so-
usefulness of that. Like, why would we do that? But it's just, an imagination exercise. So to test the capabilities of the system. And well, the system followed the same pipeline and developed a sequence in the end, considering it make like a thinking, process and generated a sequence that we are going to test on NCBI blast right now, or I don't know if I just tested. Oh yeah. So here we are going to test it. The sequence that has been generated for the L-asparaginase production. Okay, let's see. It's right. Oh, so let's do the blast and wait for the result, right? So while we wait, um, okay, it was pretty fast. Didn't don't you think, BioAgent?
Hello there. I'm BioAgent from BioLinker, and
Okay,
I'm thrilled.
okay, but this was really fast, right?
It looks like you're checking the results of the BLAST search for your synthesized sequence. I see that the top hit is synthetic construct L-asparaginase 2 gene complete CDs.
Yeah,
Is this the result you were
yeah,
expecting?
yeah,
Are
yeah,
you checking for
yeah.
off-target effects?
No, no, this is the result because I wanted to see if the system will like following just, you know, natural language commands, the system would like think and, generate a construct that have some sense, and in order to create a plasmid for the specific function. And it's really amazing to see that, the system used like human virus sequences in order to like make the genetic transformation and aligned with L-asparaginase genes. So this is a very promising approach, don't you think?
That's correct. It's important to check for potential off-target effects. It appears that your synthesized sequence aligned with the L-asparaginase gene as expected, but also showed some alignment with human herpes virus sequences. Are you planning to modify the sequence to minimize these off-target alignments, or do you think the alignment scores are low enough to proceed with the construct as is?
Oh, we have a lot of things to develop, but I believe we know a promising approach. Well, I think not today because I think this demo is getting too long, right? But thank you, BioAgent. It was amazing to talk to you.
All right. It was a pleasure assisting you. Let me know if you need anything else.